So a very good afternoon to our revered guest speakers, faculty delegates from different colleges, and my fellow colleagues. I extend a warm welcome to all of you on the fourth day of the AICTE sponsored 2-week online faculty development program on exploration of nascent pedagogical tools for updating teachers of pharmacy education. Yesterday, we had a wonderful session on journal targeting, journal metrics, and author metrics for academic benchmarking. We were updated with the important aspects of plagiarism, literature review, as well as the important points which we should keep in mind while selecting a journal. That is, we call the journal metrics, as well as the author metrics. Now, before starting the technical session, I would like to make one announcement to all the participants. We will be announcing the name of the star performer on the last day of the FDP, that is on the 16th of January, which will be decided on the basis of your regularity in attending the sessions, your active participation, and the score in the examination, which we will be conducting in the last day. So, all the best to the dear participants. So now moving ahead on day 4, we will be having 2 technical sessions today. The first session will be taken by Professor Csoka Ildiko, Director, Institute of Pharmaceutical Technology and Regulatory Affairs, University of Szeged, and the second technical session will be taken by Professor Chandragouda R. Patil. He is the Professor in Department of Pharmacology, DPSRU, Delhi. So starting with the first session, we have today with us Professor Csoka Ildiko. She will be speaking on Quality by Design, QBD, the key methodology for research and innovation to quick regulatory acceptance and market access for medicinal products and medicinal devices. So before we start the session, I would like to introduce ma'am. Professor Csoka is presently working as the Head Institute of Pharmaceutical Technology and Regulatory Affairs in the Faculty of Pharmacy, and she is also the Director General and Rector and Chancellor's Office, University of Szeged. She holds a pharmacist degree from the Faculty of Pharmacy, Albert Zent Gheorghe Medical University in 1990, a PhD degree in 1997, and habilitation 2007 in the scientific field pharmacy. She has been working as a teacher researcher in higher education since 1990 at the University of Szeged. Her research areas include nano-medicinal formulations, quality by design-based dosage form formulations, and manufacturing. She teaches and organizes educational activities at 4 levels. One is the Pharmacist Undivided MSc, PhD program, higher level vocational training, and PG specialist training course. So we welcome you, ma'am, and greetings from Lloyd. Over to you now.
Thank you. So welcome all of you, and as far as I know, and we discussed earlier that you are in an afternoon. We are here in Szeged, in Hungary, in Central Europe now. It's a morning. So I'm very happy, and I very welcome this initiative that you started this kind of internal education and self-improvement on different knowledges, whether scientific field or in case of pedagogical methodologies, because I think this is the key in higher education, to be updated in all the activities what we are doing, or teaching students, or showing example as researchers, because whatever we do in our daily work, we show examples to our students, or bachelor or master or PhD student. Let me tell you, and as you see on the author list on this first slide, that I'm very proud that that I have PhD students from your country and from your place. Yamini Kanti, she's here with me now in this department, and I'm very happy to have this fruitful collaboration with Amrish at your university. And this is, I think, this is the background why I'm here now and keeping this presentation for you. This is a collection of very subjective thoughts of mine concerning QBD. And what I collected together now, I hope that it will fit and meet your requirements or your imaginations about this topic. But we keep contact, I think, so we don't stop our collaboration by keeping this presentation and that's it. But so I think what I missed out, or there are still questions arising concerning this presentation and concerning the topic, I would advise to keep contact with each other and put your questions later in email or any other possibilities. I'm really open toward it. I start with it's a very famous researcher, Deming, who said that it's not enough, and this is my motto for this presentation. It's not enough to do just to do the work hard and to do your best. You must know what to work on, because there are many activities in life that we are doing things, but they have no sense, let me say. So it's very important that it's very good that if somebody works hard, but if it's something which has no sense or it has no target, then those efforts are going to be in a waste, and they have no use for the society, for the science, and not for the person. So my co-authors here, as far as I mentioned, Yamini is here with me from India, from your country, and two colleagues, Orsolya Lackovic and Assistant Professor Livia Adalbert. They are my group working on regulatory things and regulatory affairs in the case of development of different dosage forms. 3 parts I have in my presentation. This is going to be 60 minutes. And I divided these parts by colors. So all in red are going to be in the first part of the presentation, a kind of introduction and basic elements from where we start this quality by design thinking. Because I want to show you the way of thinking. So what is the paradigm change? Now in my presentation, I avoided to detail those lexical informations which you can find through the internet, which just because you just put on, you look in Google, and you can find a lot of things about QBD on the FDA, on EMA, on the ICH guidelines. Everything are on internet. So I will not bore you with detailing these lexical data. I would rather prefer to give a way of thinking, to share my way of thinking concerning QBD, and I want to show you all the successes and the publications what we have from the past 5 years, let's say, what we have as an experience in implementing QBD in pharmaceutical research, independently of the fact whether it's a pharmaceutical development or it's a medical device. In this meaning category, does not make any difference, because the way of thinking is starting with setting up the target product profile. So when we set up the target product profile, when we know where we want to reach, then all the requirements of the given product category are within our thinking. So this iteration and this risk-based assessment of the different steps we have to do to reach our target, it's in our thinking, independently what is the product category. So the first block is the first module is about the basic elements of quality, something about strategic management, and the risk assessment methodology. I want to show you that all these 3 are very good, that they fit each other, and we speak about the same. Whatever we speak about strategic management or risk assessment or quality management or quality cycle, you will see that we speak about the same thing, and the logic is the same. And the second part, when I show you some regulatory advantages or technology transfer advantages of implementing quality by design into early pharmaceutical development phases, because these criteria are now as an expectation put in front of the pharma industry or medical device industry. But all of us who focus and our research activity leads to a product on a pharmaceutical or medical device market, we should know this. And in order to facilitate and in order to have more success in our research And if we want that patients really do, we want our patients to really do be healed and treated by products we are developing, then we have to take into consideration all the stakeholder expectations, what we have concerning our products we are developing or manufacturing process developing. So I will show you the stakeholders in case of quality by design when we adapt it to the pharmaceutical research. And last, with blue, I will show some slides showing our article from some photos, some illustrations from our articles. But our articles are available on the net. They can be downloaded or we can send them. So any of them is interesting for you, any research is interesting for you, we are really open to our cooperation with you. So what I teach, we have here in our department here, we have 15 researchers, we have around 15, the same number of assistants, technical laboratory workers, and also we have 20 PhD students here in my department. So these 20 PhD students are taught to use quality by design in their research. So what I start and ask them to think first, what is the target? What is our motivation? How do we decide the topic? It's very interesting how we decide to have a research topic. I saw many examples abroad, okay, how the researchers decide to select a topic, and then they don't understand why they fail with publication. And they don't think that maybe the decision to start with the topic was not really good. So that means that the literature search is very important in case of PhD student. And we as the teachers, we have to show an example that first of all, before we start to do anything, we have to make a self-evaluation, an environment evaluation, and a literature research. So to find really a good target for our research. Then what are the first steps when we start a research? How do we build up our research plan? How do we prepare the steps? Do we have a plan or the steps, and do we put milestones? How do we measure the milestones? Do we have any measures at all in our research? Do we know whether the results are acceptable or not? I read and I reviewed many publications when we said, oh, the particle size of given dosage form in case of given dosage form was this nanometer and this nanometer. So what? Is it good? Are we happy? Are we sad? Is it proper according to our preset requirements or not? So these are very common problems in research, and I experience it as a teacher that the way of thinking in our PhD, in case of our PhD students or younger colleagues, it's not really clear that we have to measure. If we aim something, we have a target, then we have to measure whether we are getting closer to our target or we are getting farther, or we are staying in the same place and just making steps, but it doesn't help us to go further on. So do we know whether, do we learn from our failures or success? Do we know that we have failure? Do we know whether we have success? These are all questions when we start a research. And do we change old routines, or we use the same way? And this is a very basic question. I don't know how it is in your country, but here in Hungary, it's very difficult to change. The paradigm change, what, for example, this quality by design thinking is causing, it's not so easy. So to change the mind, to do things differently, is not so easy, but we know from great people from the history that if you do everything on the same way, you will get to the same place. So changing the mind is very important in cases. So this kind of quality thinking, this kind of continuous improvement, continuous updated for R&D and higher education faculty, I think it's very important, because what we do, as updated we are, that's the level of knowledge we give to our students, and this will give the quality of the higher education if our research activity and we are updated in research. So let me, with this, after this a little bit longer introduction, to show you some examples about the definitions. Let me show you this this short citation from Henry Ford. When we want to define quality. he said that I don't see the whole screen. That's my problem. Yeah. Okay, here I am. So Henry Ford told, I will build a motor car for the great multitude. It will be large enough for the family, but small enough for the individual to run and care for. It will be constructed of the best materials by the best men to be hired after the simplest design and that modern engineering can devise. But it will be so low in price that no man making a good salary will be unable to own one and enjoy with his family the blessings of hours of pleasure in God's great open spaces. I think it's so nice as targeting and defining quality indicators, key performance indicators for the Ford car. And this example I brought here just to show you that defining the quality target product profile, it sometimes anyway, first of all, it's very subjective. Secondly, it can be totally individual, and it can be very different factors. But without knowing this, and Henry Ford knew how to start, and this was his aim, and he achieved it, I think, because he knew it. And then something about this is also not a joke, just an example from life that also Henry Ford said that the customer can have a car painted any color he wants as long as it is black. So he didn't pay attention on the color as a key performance indicator. So the color of cars for him was not included in the target product profile, or the quality target product profile. And the lower place of the slide, you can see two laptops here, two notebooks. And then it was an advertisement running here in Hungary, and it was about a chat between two young people, and they were discussing. One of them, he wanted to sell, and the other one wanted to buy a notebook, and they were discussing on the processor side, on the ROMs, on the technical parameters of the laptop. Finally, the guy decided to buy the laptop from the girl. And finally, when they met personally, then and the girl took out the laptop from her purse, then it cleared up that the laptop is pink in color. So finally, the business was not, the deal was not managed, because the guy said, okay, he didn't have in his mind to ask about the color of the notebook, but it was not in his target to have a pink laptop. So there are places when the quality target product profile, or even in case of a medicinal product, maybe the color can be important. I think none of us saw a green colored capsule or tablet. Maybe there are, I don't know, but no. So there are some colors which they simply don't fit to different things. So how to define quality? When we come to this question about the definition of quality, then we know from the literature and from the quality assurance scientific background that there is a technical quality definition to meet the requirements, or given requirements, or suitability for purpose, or meet a standard, like in case of pharmaceutical products, or to reach customer satisfaction. But if the quality definition is to reach customer satisfaction, we have to know who are the stakeholders, what are their preferences, and we should get some feedback from them in order to meet these requirements, or to meet this customer satisfaction. So in a competitive market, it's very important to reach customer satisfaction. I will state that both quality definitions and focusing on both quality aspects is very important in our case in pharmaceutical research, because in the first case, when we speak about the technical technical quality issues, then we have to say that quality does exist independently whether the customer realizes it or not. There are some technical, like microbiological impurities there won't be any stakeholder requirements it won't be a customer satisfaction question. It's something technical which should happen based on professional standards. This is out of question, and it's not a question of being aseptic in case of ophthalmic preparation. It's not a question of making business, whether I'm satisfied with it or not. And the second part is much more and more important nowadays, because we work in a global pharma market and a very competitive market. And the society globally, people, they are changing, and very rapid change you can see in individualism on selecting among the products by own decision. So we have to know, and we have to contact our stakeholders, whatever we do. It's the case in case of higher education with students, with the parents of the students, with the society, with the professionals. So then we have many stakeholders. We have to make discussion. The scientific literature about this says that the healthcare now is moving toward the co-creation. Marketing people, they know this term. It's an old term, co-creation. But in case of healthcare services and healthcare products, we more and more move to patient-centered care and co-creation type services and product development. I will not use time about the history of quality assurance, but it's very important to see that to reach from simply quality control, a retrospective way of thinking, to go to total quality management, and from testing to designing, it's a very important paradigm change. And this paradigm change is the person who we have to mention as the first person to develop and to deliver up the theory of QBD is Uran. And this person, he went from Romania to United States and those days, and he wrote the first scientist who realized that quality planning is very important, and not only product in case of developing products, but in developing processes. And all these product design and process design should be very important. He was the person, just let me share with you, who sent a message to Europe when this was before the Second World War. And in Europe, in case of quality management systems, we started here in Europe with ISO system, with ISO standards. And Uran sent a message to Europe that you can develop these ISO standards, but if you are doing things in the same way, you will reach to the same place. So doing something very precisely without questioning whether the aim is proper or not, or without thinking on the fact that the aim can change and continuous improvement is necessary in a changing environment, in a changing society, and in a competitive market, then just do it. But you will be in the same place, doing things very standardized in a very standardized way. Okay, ISO systems, they developed a lot during the past few decades. So now it's not the case, but on those days it was really real critics. So Uran says, and these we call Uran basics. There are very good books on internet about Uran way of thinking. So who is interested in it, I can advise you to read those ebooks. So first of all, he said customer focus. It's very important to define stakeholder needs, plan the quality, and then to set up key performance indicators to control check the process and measure, and celebrate if you have success and improve if you have failures. But the importance of Uran's thinking that if anything happens, success or failure, to know it in time. So this kind of in-process control thinking, milestone thinking, iterative way of thinking, risk assessment thinking, and not setting up the requirements and going directly in that way and without any measuring, it won't make you competitive and won't make a good quality result. And this is true even nowadays when the requirements or the market expectations, they change very, very quickly. So if you don't have a way of thinking that if you decide something and you set the target, but within you are within the process, you are not thinking and not looking around what can happen, what are the risks, are there any changes in the environment? So if you are not aware of changing environment, then finally when you reach your goal, it will be old, or it will be it won't be competitive enough, maybe, because other person. This was after the Second World War, Japanese car industry, automobile industry, and their competition with the American automobile industry. So the Japanese cars were winners due to their quality thinking on continuous quality improvement. And this was the opposite, totally opposite what the Americans do. It's also very interesting. There are stories also. So who is interested, I can advise to read about the automobile industry competition between Japan and United States. So the third point of Uran's trilogy, is called the Uran trilogy, is that quality cannot be tested into a product. The target should be set up, and then it should be designed. And this is the way of thinking of Uran, which was implemented and adapted to pharma industry. So it's totally an industry independent logic. QBD is not a pharma, finding, and not a pharma idea. It comes from a general quality word. And this is how we translate into pharma. Customer needs, we said, this is the quality target product profile. The product features, we say critical quality attributes. Input variables, they can be material or process variables. Control strategy, design space there. So Uran said that we have to set up dominant variables, time dominant, component dominant, worker dominant variables. So his way of thinking was taking into consideration within the process, whatever can happen, it will. So we have to count with them. Some of the variables or some of the happenings we cannot avoid. So the aim is not to avoid, but to count with them and implement them, or to have a plan. If they happen, then what we will do. So to evaluate all the possibilities from the worst case to the best case, but always prepare for the worst case. And then only positive things will happen during the process. So we need, finally, to conclude this, we need both quality aspects in our thinking, technical natural quality, and also customer expectation and satisfaction. So when we state that we do pharmaceutical research, then we cannot leave out the stakeholders' requirements from our thinking. There are many, and I will not, I don't want to criticize any researchers, but as far as I think you also see, like me, that and globally in the publications, there are many publications stating that we are doing pharmaceutical research. This is pharma development. This is a potential medicinal product can be from this. Even basic researchers, they state that. Yeah. But if they don't take into consideration what, for example, regulatory authority requires, and they are using, let's give an example, organic solvents, and they do not count with the ppm of the remaining solvent in the final product, they cannot say that, okay, we are doing pharma research, because never it will be on the market, because a regulatory agency won't let. Or if nanopharmaceuticals or liposome preparation, any kind of complex or biological drugs, if the early stage of thinking, they don't have, okay, so what will happen if the regulatory authority won't let this composition to the market, then I cannot say that I'm doing pharma research, because I don't take into consideration, and in my thinking, I have no idea about the KPIs which are set by the regulatory authority. If I don't have in my mind that pharma industry, cost effectiveness, time thing. The number of steps in manufacturing process, the time needed. So there are several indicators pharma industry has in its mind as using it for the decision whether they start a product manufacturing or not, if they put money in it. So leaders, when they decide, they have these kind of questions to the researchers. Okay, oh, you have 15, so we won't find an industrial partner to manufacture our developed product. So we have to think on it. And last but not least, the patients. So if we don't take into consideration what patients prefer, then we can have excellent product. The regulatory authority will pass all the requirements what they put in front of us, but there won't be people to use it because it's not convenient for them, or simply they cannot adjust their daily life to the dosing frequency, or there can be many aspects. So if we speak about targeting and good targeting and good, for our research activity, then it means that we have to know our stakeholder requirements. Another example, just to close to finish for the first part to use our time good, I just show you the strategic planning cycle. Is the same like the quality cycle: plan, do, check, act. Set up the goals, set up the desired outcomes, to set up strategies to reach, measure where you are, and results, evaluation of the results, and then how can we improve. They call it strategic management. It's the same. I'm the Director General for Strategy at the University of Szeged. I work for the Rector and the Chancellor. So it's the same. I use my knowledge from quality assurance, what I learned as a pharmacist, or I learned at the university. So teaching quality assurance, teaching quality management and quality management tools, I think it's a way of thinking we should teach for our students. We don't teach quality management, we teach design thinking, we teach the way of evaluating life around us and set up target. Ask your students whether they wake up every morning, whether they do plan their days, or they just wake up, they spend the day, and before going to bed, they said, okay, what I did I do anything useful today, or I just use my day? So normal people, even when they wake up, they think through their day. They have targets for the given day, and before going to bed, then normally we think through, okay, what I did today. This is a very simple way to teach students on a way of thinking, or strategic thinking and quality thinking. This is the same. Stakeholder evaluation. You have to decide for who you want to meet, whose requirements you want to meet when you plan something. You can plan anything, you can have great plans, but without a precise target, your plan won't be useful because it won't fit exactly. One more example from the literature: risk-based process management. The same. It's not written in this figure. It's not as a circle, but the same. Identify what kind of hazard can be, determine, then assess, do some action, and evaluate. The same continuous quality improvement way of thinking. So we cannot stop thinking whether we can do things better and better and better. So this is not a static procedure. So independently, whether it is an individual process, let's say a research project, or it happens at an organization level when you design the work of your research group or the work of your department. So independently, whether you work in a single project or your single individual personal life, self-evaluation, environment evaluation, a kind of SWOT analysis. There are different analyses. I also teach my students what does SWOT analysis means. Measure your strengths, weaknesses, opportunities, and threats. And then how to evaluate the environment around us, how to make literature search. This is a literature search. Does it have any sense to do research on that topic or not? We should evaluate first. And then when we found the gap where we have to go because there is no research on that area or there is no product on that area, and we read in the literature that people, they would require, there are many examples when we figure out, oh, this will be a good target because, and you quickly check around the literature whether it's needed. So it's very important after evaluating the situation, then to be target specific. So the first question when we start, and now we turn to green, to the middle part of the presentation. So who are the stakeholders for our research activities? This is what we have to look for. What is the market we want to reach? And the question is whether the quality management is only the business of the pharma industry. My answer is that no. If we want to stay only researchers and we don't have the wish in our hearts to heal patients and to have products on the market somehow, independently that this is a food additive, this is a medical device, this is a pharmaceutical product, whatever, but what keeps people healthy or help them to be healthier or to recover and to show and to develop treatment for patients. I think this is in all of arts, all of us, we have in our hearts as a wish, as a motivation of our work. So if we say this, that we cannot stay in the research, closed in the research, but we should go further on, and we should go what patient wants, what the industry requires, and what the regulatory agency wants us to reach the global market as soon as possible, because that will give a success for researchers. I have publications. As I'm getting older, they will increase as I have more and more colleagues and we work together. Yes, now the number of publications, they increase with time. That's normal. But what I'm the most proud of are those patents I have, which mean that there are products on the market and there are some people using it. So that gives me I think the best feeling that it has sense what I'm doing. So if we want this and we have this wish in our hearts to do something useful for the society, I think that QBD way of thinking can help us to have this. So I think this is our responsibility to learn something about these regulations. So okay, how to make it? So these are the term innovation ecosystem is very often used nowadays, but this is our innovation ecosystem. We are, as an academia people here, we have the pharma industry partners, we have the regulatory bodies to work with, and the focus on the patient needs, but also together with the patient, as I told before, as a co-creation, which means that the patients are as well in the part of the ecosystem, and we don't develop anything without them. But it's very rare, let me tell you, it's very rare that we ask the patient. For example, in case of pharmaceutical technology, do we really ask the patient what kind of administration route do they prefer? Okay, we say, and also I have heard many lectures that yes, the patient compliance is better if we use Perros possibilities, and yes, and we developed the transdermal patches. They have very good patient compliance. Yeah, this is our hypothesis, and in most of the cases, it's right. Yeah, the patient adherence and compliance is better in case of transdermal patches instead of taking Perros 4 times daily tablet. That's true, but do we have measures for it to a different patient population in a given disease or whatever? So when I started 5 years ago to think on this, then I realized that we don't really have data on it. So evaluating patient needs and patient attitude and this kind of customer expectation thing in pharma industry, and then I realized that there are no even nation specific, we don't have We didn't have, for example, osteoarthritis is a very common disease globally and even in Hungary. And I tried to find out that Hungarian patients, what is their attitude towarding this disease? What do they prefer? What to develop for them? But I didn't find any kind of data. So we developed, one of my PhD students, now we published in Journal of Musculoskeletal Diseases. And now we have a publication, we have a questionnaire. We developed a questionnaire, we adopted international questionnaires about the health-related quality of life. And this disease-specific questionnaire, now we gave us the answer. So what should we develop? Exactly, what are our patients' needs? So this slide shows only the technology readiness levels. This was developed by NASA. I don't know whether you knew it. It was developed in the 1960s by NASA, and they evaluated and they realized that they should give different management methodologies at the different levels of readiness. This is a technology readiness levels. I won't detail it. I just brought it here just to show you that this is adapted to pharma developments. So till the prototype, till TRL4, it's research, and from them it's development. And at the universities, generally, it's true that we stay in the research phase, and we don't go further on. We don't pass the fourth level to go for the development. So this part, this right part of the whole process should be facilitated to reach, to get more products on the market. So the message one here is a conclusion of this, and I'm sure you know it as teachers, researchers, but I want to emphasize that we have to teach this also to our students. So what is your aim? Target product profile, set up hypothesis, plan your research steps, define the risks, define the indicators, evaluate the results, and develop the method. This is QBD itself, but this is a way of thinking. This is the same like the plan, do, check, act, but it's adapted. And I think my last slide to emphasize the importance of setting the target, but this is, I focus on this maybe too much, but I think this is the weakest point. This is the weakest point. To set up a wrong target can destroy lives. We have PhD students here for 4 years. 4 years to reach our scientific results is not too long time. So it's very risky. For 4 years for a PhD students, you really have to set up good target as a teacher, as a leader researcher. This is Alice in Wonderland. I don't know whether you know this story. We know it here in Europe, Alice in Wonderland, and this is the short story from this, that one day Alice came to fork in the road and saw a cat in the tree. And she asked, which road do I take? Where do you want to go? Was the response. Oh, I don't know, Alice answered. So then, said the cat, it doesn't matter. So if you don't know where to go, any roads will take you there. So coming back to the pharmaceutical development, I won't detail this. This is the difference of the approaches of the current approach and the QBD approach. I think this is well known, but this is the way of thinking that you preset parameters, systematic thinking, design thinking, and taking all the possible problems or challenges on the road where we are going to reach our target should be taken into consideration. Like a careful traveler, or when you drive a car, you always look around if anything comes in front of you. So this illustration is also, I think, known that if you have all the excipients, all the manufacturing process meeting the specifications, and you have fixed process parameters, fixed in everything, then still can be problems. And specifications simply, it won't be enough to a good product quality. That means that if anything changes, or the excipients, or the manufacture of the excipients, you know the regulatory requirements. I think you know well that if anything changed, even the equipment or the excipient producer or anything, then you should apply to the regulatory authority that change type 1A, 1B, 2A, etc. So if anything changes when you have a specific process description to the regulatory authority, whatever changes, you have to apply for the new acceptance again. So all the changes should be regulatory approved. And this is very time consuming, and it's very, difficult, and needs a lot of people. So with quality by design thinking and applications and common technical documents given to the regulatory authority with the thinking and QBD administration, it's much more easier because you define the circle, the design space, and the design space makes you the possibility to move within those borders. I have many examples, but I don't want to use the time for this. So the advantages, and you know these, I think this kind of ICH guidelines, writing down and describing the QBD methodology and the aims. So for sure it's saving time and cost to develop high quality products, and it helps you to get closer to the market. This is the Uran's logic. Do you remember Uran's trilogy? This is the Uran logic in pharmaceutical industry. And this is quality by design. This is the official scheme. And this is written in the different regulatory, the different guidelines by ICH. Let me ask the question, these guidelines, or this, because this is for 20 years, I think, quality by design, and these guidelines, some of them, it's 2009, so they are quite old. Do these have any impact on our research? Just this is a question for ourselves. Do we know what they speak about, and does it have an impact on our way of thinking? Hopefully yes, because I think all of you who are here, you are here because you want to learn. So I think that you know this and you are updated in this. But you have to call attention to this, to our students as well, and also to our younger project. So about QBD, some notes about the regulatory aspects. So in 2011, the EMA and US FDA started the joint pilot program for this. This is on the website of the EMA. I won't detail this because you can find it on the internet. And as I started my presentation, I don't want to use those materials which are already on the internet, because you can find them. As a student, I was really bored when my teachers, they started to read up from the books, because I thought that if I have the book, I will read it when I'm at home or I have more time for this. But for the lectures at the university, I wanted to be on those lectures only, which were interesting enough, because the knowledge of the teacher was interesting enough and deep enough to teach me, and I was rather interested in the way of thinking of the teacher instead of the lexical notes, because lexical things can be found here. So what is important for this? That like ICH is globally valid for all the countries in the world. Also the QBD documents and the harmonization documents are really important, and this means that QBD can be applied all fields of pharmaceutical research, and it allows us wider collaboration at early phase of research. So whether we independently which country or which continent we are speaking about the same way of thinking, and this will lead really the success. Yes, this is also from the website about the meaning of the QBD. And also I have my presentation available for you. So if you want to use it or you want to get it, that's really, for my side, I give it to you. This is the FDA design control perspective concerning the QBD. what is important from this slide only? That design input, design validation, design transfer. So what they look at this type of review, how did you evaluate it, the present, the literature, how do you describe the design, what is the knowledge behind, whether your procedures or your composition design is knowledge based, and there are evidences behind your decisions. So this kind of regulatory thinking is not anymore the same like it was before, that the requirements, okay, meet the requirement, yes, no. If no, failed. If yes, okay, passed. But this kind of evaluation needs, anyway, it needs much more knowledge from the regulatory people. So this, and I'm very happy that a new scientific field raised up, regulatory science. And I really do believe that regulatory science is a real interdisciplinary science, and a very interesting one, because you have to combine the knowledge from all the scientific fields, analytical chemistry to pharmacology, toxicology. In a regulatory science, everything is integrated. So it's an integrated, multidisciplinary and interdisciplinary science. And regulatory authorities, they won't be anymore simple administrative, bureaucratic persons, but they should be scientific, having scientific background, and more, and I'm very happy to see this, that more and more people, they have scientific background, and more universities, they are involved as experts with the regulatory bodies. Something about the medical device development, which was just started this in our institute here, but the logic of QBD is totally the same. But this, the end of May, it was postponed with 1 year, the new MDR. But now it's told that now they will start it. So the May 27th of May this year is going to be the MDR day. So these questions, which products can be stay in the market, and we are preparing here in the institute at the university with a clinical background, because we have a clinical center here at the Szeged. We have 31 clinics, so the total portfolio of healthcare we have here behind. So together, scientists with biologists, chemists, and all immunologists, and all scientific fields now, we put together research groups on medical devices, and also we have the clinical background for the clinical evaluation of the medical devices in the given category, which becomes now compulsory from this date. But here is a white paper. It's from a white paper for medical devices QBD. The same logic. So that's why I started my presentation, that independently of the fact what is the product type, because if you put the requirements valid from that given product type into your process, into your design, then it won't be a surprise. And it won't be at the end of the process that you know which product type you are developing, but you have to know it when setting up the quality target product profile. And here I come to the blue titles. And this is our publication from 2018. You see it on the slide. We made an extension of the quality by design concept, which was delivered, which is valid and compulsory for the pharma industry, and it's given in the ICH guideline. And we made an extension, and we call it now R&D QBD. And this R&D QBD, we set up this. You can find this article on the internet. You can download it. That means that we put some places. This what you see in red, some extra steps within the QBD procedure, because research should be more iterative than an industrial. So this first, the new element in this R&D QBD. Is it comes here? Oh, sorry. To start with the knowledge space. First of all, to see what is this is we call pre-formulation design space. not to have design space only at that step when we are making the product and the manufacturing process. But first of all, to think what are the unmet clinical needs. It's very important that they evaluate this, to select the API, and depending on the API, whether it's biological, herbal, natural, or synthetic, we have to evaluate the biopharmaceutical aspects. Here we have to select the administration route, and here we have to evaluate the dosage form possibilities. And this is the pre-formulation study design. We have to think first what to treat, with which type of drug, what according to the drug's biopharmaceutical aspect, to choose an administration route. And then if we have the administration route, then go back to the next question, what should be the dosage form? And when we are ready with this, then we can start further. So this is the early phase when you have to think. And just to make it that this is a valid way of thinking, the evidence behind this is that the WHO started in 2013 to put and to finance especially those research and development projects which go for the treatment or go for treatment of the special diseases. Here are listed osteoarthritis, diabetes, dementia, antimicrobial resistance. So they have some, you can check WHO website, and also you can see that what kind of challenges are still on the earth to be solved. So those projects are going to finance and are financed now by different grants which go for these unmet clinical needs, not about something which is already discussed many times. The European Medicinal Agency made this PRIME. I don't know whether you heard about this. This PRIME, this is a fast-track procedure and regulatory pathway for those researchers and those products which treat these kind of unmet clinical needs. And that's why they have for orphan drugs, they are different pathways. And this is the list, this is the evaluation. We don't detail it, that which medicines, what are the key features, when can you request this kind of PRIME. So this is a fast track if you really have a research which is very, very urgent and meets unmet clinical needs. So this is our first question when you want to set up a research activity. So what to research? First of all, to see that globally, what are the unmet clinical needs? This is a statistics that how many were granted, and you see the most of fast tracks were granted in case of oncology, second one hematology, hemostasis, and infectious diseases, neurology. So these four are the drugs, dosage forms, and products on the market getting fast track because they fill the requirements of unmet clinical need. The second step in the new R&D QBD is which differs from the original one, which is in the ICH guidelines, is that when we define the quality target product profile, here it's very important to make a stakeholder analysis. And here are the industry as stakeholder, the regulatory, and the patient. And I added one more, the social environmental responsibility. This means the green technology, the economic aspects, so the sustainable development goals, which are globally important for all of us, to use the green technology as much as is possible, economically efficient methodologies and products suitable for scale-up, automatization, etc., etc. So then when we start our research, then in the middle column, you see the regulatory aspects. We have to take into consideration what is the targeted authorization procedure and product category. Is it an original drug? Is it a generic? Is it a biological? Is it a medical device? Is it a food additive? What? Because the requirements of the regulatory bodies will depend on the category you want to put it. So, but this is a decision at the first time. I was involved in many researchers, believe me, there were very good ideas. They put a lot of components in it, nice composition, very nice. But finally, when the industrial partner decided to put it in a medical device category, then they realized that there are components in this composition which is not allowed in case of a medical device category. So two chances: or to weaken the product to subtract some of the components which were not allowed in this category, or to put into the pharmaceutical product, medicinal product category, which is much more money to put costs on. So finally, our research, our product failed. However, as a researchers, we did a good job, but there was not this how to say, the discussion was not proper with the industrial partner. So what category you want? So deciding what the category we want to run for, it's very important. It's a part of setting up the quality target product profile, the category, the regulatory category. Then to set up the requirements for the excipients, toxicity, hazard, evaluation of different, here many nanomedical medicines, nano everywhere. But it's very important to evaluate the accumulation, the toxicity aspect, everything. But as a first step, not as a second, not the last step, not when we want to put in the market. And we are very happy with our research, but we don't count with the possible failure and the regulatory authority due to the toxicity aspect. And then last but not least, the patient. For easy administration, for comfort, for adjustability to daily routine, not to feel patient, not to feel sick, proper dosage regimen, and whatever. So these are the three stakeholders. When we set up our research plan, it's very important to get off. So my time is almost over, and maybe over. These are some examples here from the literature only, that how is important, what is the EMA definition for the pharmaceutical design of a drug. These are which are important in case of quality target product profile, composition, dosage form, route of administration. These are what I put in the pre-formulation design, because EMA wants these data. So we have to preset these requirements. Some about the patient-centered pharmaceutical design. There are many articles concerning this. You can see that what are the problems for if you prepare geriatric preparations and the older patients, they have swallowing problems. You have to count with this. So this is targeting to the patient population. You have to segment the market. Market segmentation is also important in case of pharma research, like in case of marketing when selling something, because finally we want the patient to reach this. Okay, do we prescribe what patient prefer or not? It's a very interesting question. That what I mentioned before. Tablets are the most prevalent and preferred dosage. What the different dosage forms? What are the patient preferences concerning this? It's important to know. Shared decision making is also a term. I made research on this as well. In case of rheumatoid arthritis, what patient like and what not like. If you develop a product for this disease, you should know what patients suffering from this disease think or they expect. Okay, oncology patients. What oncology patients prefer? Then about the administration route. Intramuscular is better than intravenous, or subcutaneous better than intramuscular. It's also a kind of patient preference. So design space is about adjusting and merging together all those stakeholder expectations, what we have from manufacturer side, from industry, from regulatory, and from patient side. And this will give us those plots and that era we can think within it. Here are also the attributes, the route of administration, dose frequency. This is what I mentioned. One thought: quality by design can be used in case of all scientific field: analytical chemistry, pharmacology, toxicology, technology, regulatory science, whatever. And I collected some articles about implementing quality by design in chromatography. I won't list it, just an example. Analytical methods, quality by design approach in case of developing an analytical methodology. Quality by design in case of extraction, herbal drugs. So quality by design is not only for pharmaceutical technologies and product developers, but it's in all kinds of scientific field. You will find articles, and please look for it. QBD for herbal drugs, articles, some articles about this. So here the message is, and I'm close to the end. The message is here that in all fields of you are having research, QBD can be implemented. Here, this is a flow chart on the right side of the slide. What we are using here. Okay, just if you have 2 minutes more patience. Sorry for being longer, but let me just show you that how we set up in case of this is an oral delivery of peptide drugs. We deal with peptides. With nanocarriers, nanomedical systems, and also antimicrobial peptides and alternative administration routes like pulmonary and nasal administration route, but also with peroral administration routes. Here you see just an example from our article, how we set up the quality target product profile. This is the risk estimation matrix. We have a lean QBD software. We bought a software to make this matrix about the availability of risk. And with this risk estimation, here is the Ishikawa diagram. This is the also the polymeric and lipid nanoparticles we are developing. What are the factors influencing the quality of nanoparticles? This is a part of risk-based assessment. Here is the design space, the design of experiments and design space establishment in case of polymeric nanoparticles. Here you see the Pareto charts concerning this. What are the most effective, most risky, or most affecting parameters in case of polymeric nanoparticles? Here you see a flow chart and risk assessment in a case of a new peptide formulation for oral drug formulation, how we should decide what is the decision tree, what is the flow of the process. Here is about an antimicrobial peptide, a QBD-based pegylation design. Even for one process, we can set up a QBD methodology. And when we have the pegular procedure and the pegylated antimicrobial peptide, then we put it in a nanocarrier system. Here we are now. This is still a picture about the Ishikawa diagram about the quality of chemically modified antimicrobial formula. What are the effects affecting the quality? What are those critical attributes, material attributes, which will be critical quality attributes? This is a solid lipid nanoparticle for Alzheimer's therapy in nose-to-brain delivery. What are the CMRs? What are the CPPs? What are the highly risky factors? This is our evaluation by this software. A liposome formulation, how we do it, the risk assessment, and how we do the design of experiments here. This is also an example, the risk estimation matrix, the Ishikawa diagram in case of a liposomal formulation. And one interesting point I want to show, QBD methodology can be used not only in case of analytical chemistry, pharmacology, whatever field, but also in patient therapy. Here is the quality of life Ishikawa diagram. So if our aim is to reach quality of life, these are those elements: mobility, self-care, usual activity, anxiety, depression, pain, discomfort. These are the main factors affecting quality of life according to international standards. So we can develop Ishikawa and we can develop therapy with QBD methodology, because with this risk estimation matrix, we can define what are the highest risk in disclosure or patient inadherence. So finally, the summary, it's very important. As patient-centered care, co-creation in healthcare and in pharmaceutical development is nowadays really a hot topic. Everything goes for the patient. That means that without asking, without making questionnaires, without making literature research on the given disease we want to treat and about the population in that given disease, what are the high risky points, then we won't be able to handle this. Therapy shifts toward novel dosage form, more complex drugs. You see, our compositions are not such simply one excipient, one API, and that's it. There are more and more complex compositions, so quality by design helps us the interrelation of different factors to be able to evaluate. Industry needs for quality by design because of the cost, innovation, and quick market research. If we are not researchers, partners for industry in this, we won't have success as researchers because they will look for researchers and universities and research groups which know what QBD is. So this is a kind of competitiveness, a question of competitiveness. And regulatory, they need this also. So I'm really thankful for your attention. Hopefully only a few of them fall asleep like in the photo, but and I think I hope that I could transfer my way of thinking on always being a fair, looking like, I don't know how it is English, whether I tell it in a proper way, suricata. Do you know the animal of suricata? Which there is a group of small animals, and there is always one who is standing and looking around what happens and calls the others whether a danger comes. So we have to be like suricatas in the animal world, looking around and looking, because those will be winners who are able to detect the changes around us, and those will be competitive and winners. So thank you very much for listening to me.
Thank you so much, ma'am. You're absolutely right. We must be good listeners and always be attentive, just the animal which you named. So, ma'am, we have one question from Mr. Deva Prasad Ghosh. He's asking, should all the designing of research be always patient specific?
I think patient group specific, but not patient group specific in a meaning that a matrix of patient group and disease.
Okay, right. So it should be a mixture or matrix of both.
Mixture of matrix of, yes.
Okay, right. Thank you so much, ma'am. Just a minute, I'll just share my screen. If you can kindly stop sharing your.
Yes,
Yeah,
I'm doing.
yeah. So thank you so much, ma'am, for an enlightening session on quality management, strategy management, and QBD in pharmaceutical and medical device research. Thank you so much. And please accept this e-certificate from our side.
Thank
We will
you.
be mailing you shortly the certificate. Thank you
Thank
so
you very
much,
much.
ma'am.
I
Thank
wish
you.
you good work for the rest of the training course.
Thank you
Goodbye
so
for
much.
everybody.
Yeah, thank you so
Goodbye.
much, ma'am. Bye bye. Now moving ahead to the second technical session by Professor Chandragouda Patil. He will be speaking on hybrid learning for practical classes, simulations, and open source LMS. So, I would like to first introduce Sir. Professor Chandragouda R. Patil is presently working as the professor in the Department of Pharmacology, Delhi, Pharmaceutical Sciences and Research University, that is DPSRU, we call New Delhi. Prior to this, he has worked as professor in R.C. Patil Institute of Pharmaceutical Education and Research. He has several publications in journals of international and national repute with an H index of 22 and 1382 citations. He has been awarded one patent and has filed three more. Sir is also the author of the Animal Simulator published by Elsevier. Dr. Patil has guided till now 55 postgraduates and 5 PhD students. He has been invited as a resource person in more than 75 national level conferences. He has received several awards. Some of them to be named are Innovator Teacher Award in 2018 by MHRD Young Scientist Award, from CRH Ayush in 2017, and in 2007 at 6th World Congress on Alternatives to Animal Use in Life Sciences held in Japan, and as well as the Career Award for Young Teacher by AICTE between 2009 to 2012. We welcome you, sir, and greetings from Lloyd. And over to you, sir.
Yeah, thanks a lot, madam, for a nice introduction. It is a privilege for me to share this dais twice, the second time now.
Yes, sir. It is in fact an honor for us also, sir.
Yeah. So last time I was talking about introductory aspects of this pedagogy. Okay. Just now, audience has listened to quality by design by an expert in medical devices, I think, and it was really related to research activities in academia and industry. But when we think about nascent pedagogical techniques, which is the topic of this particular FDP, there is also something related to quality. There is also something related to design. And with the tempo that earlier speaker has created, I would like to continue the same concept, similar kind of seriousness in the field of academics now. Okay, let me share my presentation. Now let's see. Last time when I talked on this particular platform, I had talked about blended learning. Okay, and now in today's session, I'm using the word hybrid online learning. So this is not something like juggling with the words. Actually, these words are being used in the pedagogical research. Okay. In addition to my introduction, I would like to share with the audience that I had developed some interest regarding the pedagogical aspects where I myself faced problems in laboratory teaching. Okay. Particularly when, as a lecturer, when I was trying to demonstrate to my students some experiments in pharmacology, always there was some misunderstanding on the part of students, and it used to become very irritating for me. And through that experience only, I thought that something should be prepared for the students, which will be enabling them to repeatedly see the demonstrations, perform something on the computer screen or mobile screen, and then that will be helping them to learn better about the practical aspects of pharmacology at least. Okay, so that was the initiation uh, in my personality because of the problems which I faced in the wet lab pharmacology experiments. Okay. After that, I came up with the software which was available in the market as Xcology. Along with me, there was another professor from Pondicherry, Dr. R. Ravindran, who had developed, means he was before me, he had developed XPharm. Okay. So whoever is a teacher of pharmacology here out of 106, he must be aware that earlier these softwares were there for teaching experimental pharmacology. After that, Elsevier company came in picture. They asked for the copyright from us and they took our help. Again, we developed the entire software with Elsevier company, and that was available throughout the world for last few years now. So that was the way I was connected with the field of pedagogy. That is the reason why I chose to talk in today's session also, particularly about practical classes. Okay. All of us know that because of Corona, the entire pharmacy fraternity, particularly teachers and students, they have been compelled to go online. Okay, this is something very new for us. And that is the reason why a FDP like this and multiple FDPs throughout country are being conducted to sensitize the teachers regarding strengths of this online teaching learning methods. And then this is for the first time that at such a wide scale, people are talking about pedagogy. Okay? Otherwise, if we consider any earlier decades, pharmacy teachers had never come across the terms like pedagogy or pedagogical research. Okay? But this is quite important. And in that now the two words are coming up. One is blended learning, another is hybrid learning. Okay? So blended learning now is getting the name of hybrid learning. Okay, friends, before going through this actual presentation, let me share with you what I'm going to include in this particular talk. The agenda for this present discussion will be learning objectives. Okay? And I am sure that within this last 3-4 days, at least someone must have talked about learning objectives. Okay? Then I'll be telling you regarding some developments which have happened in the field of hybrid learning. Then we'll be talking about a new revolution that has come into picture, that is open educational resources and learning management systems. And at the end, I'll share with you some resources which are specifically useful for conducting practicals in pharmacology and related subjects. Okay, so this is what I want to share with you in this next 1 hour. So let's start with learning objectives. Okay, I would request a couple of teachers from the audience. Is there someone from pharmacology here? A teacher from pharmacology?
We'll have to find out.
Yeah.
Please, can the teachers raise their hands if the ones who are from pharmacology?
Yes, someone of them can just talk in between.
Yeah, we have, so there you can see their
There are
raised
two
hands.
participants are
Yeah,
there.
yeah, yeah.
So can they unmute and tell me something about learning objectives? Like have they come
Okay.
across something like learning objectives in their
I'll tell
syllabus?
them. Preeti, can we unmute them?
Any one of them? Yeah, one or two of them.
Preeti?
Professor Vivek, can you share something regarding the learning objectives? Have you come across learning objectives in the syllabus of PCI?
Sir, actually, in present scenario, like we are taking online laboratories only. Like we are recording the things, whatever we do in labs, and we are showing
Right,
to the
right, right.
students.
Yeah, that's very nice. But my question to you is, do you know that there is something like learning objectives which are given in the PCI syllabus?
Yes, sir. Yes.
Okay. So have you come across any learning objectives for practical classes particularly?
So in PCI syllabus, sir, if we see that in syllabus itself, they will be providing us with few objectives.
Right,
Like
right.
yes, sir.
Okay, thanks a lot, Vivek, for sharing this information. Friends, actually why I raised this particular question is because in our PCI syllabus, we are having learning objectives only for theory classes.
We don't have a practical, sir. Yes.
Right, exactly, exactly. So this is really an unfortunate situation. See, because my question here is, can we use the same learning objectives of theory to the practical classes? Or there should be a separate set of learning objectives for practical classes?
There should be set of different
Right,
objectives for practicals.
exactly. So this is where, unfortunately, we are lagging behind.
Yes, sir.
We have not been provided with with specific learning objectives for practical classes. Okay.
Yes.
And I'm sure that out of these odd 100 teachers who are there, no one of us has tried to pin down such kind of learning objectives for our practical classes. Is there anyone who has tried to pin it down? Like we should have a separate set of learning objectives for practical classes. I'm sure very few or none of us might have done that. Okay? At least I had not done that when I was conducting practical classes. Okay. But friends, these learning objectives are very much important to direct. Yeah, please. Is there anyone who wants to talk regarding this?
No, sir. Just by mistake,
Okay. All
somebody
right. No problem.
unmuted.
No problem.
Yeah.
That's fine. But if suppose you are interacting like this to my questions, I'll be very happy, and that will be really directing me how to go ahead. Okay. I request the organizers to enable people to talk in between if they want to share the things.
They are already enabled. They can unmute themselves. Sir.
Yeah. Thanks a lot, man. Thanks for this.
Yeah.
Okay, so as Professor Vivek has shared with us that in our PCI syllabus, there are no learning objectives for practical classes. Okay, maybe that is the reason why we are feeling that we can teach practicals online. Okay, otherwise there must have been a great upheaval against this kind of directives. Okay, now a simple question: can we really conduct practical classes online? The answer sure is no. None of us will claim that he or she is efficiently conducting practical classes online. That is impossible. Right? But friends, if we look at developed countries where online education has become sufficiently a normal thing, or people are even looking at learning objectives for practical seriously, there they have got a very nice set of learning objectives, and then they're sure what they're saying by a particular line in those learning objectives. Okay. Now, as there was nothing available in the PCI syllabus, I tried to search for learning objectives for practical classes using internet. I visited multiple websites of foreign universities, top level universities, and I came across one website of Leicester University. In the website of Leicester University, we can find a set of learning objectives for practical classes. Okay, so I'm trying to share that particular screen with you. I hope this is visible to all.
Yes, sir. Yes, sir.
Yeah, thanks a lot. So I request all the participants to go through this particular website after when I finish this lecture, or whenever they are getting time today. Just have a look at this particular website. Okay. For practical classes, they have prepared very nice learning objectives, and we can just copy them from copy these learning objectives from them and keep it with us with our learning teaching plan. Particularly, that will be very helpful to design our practical classes. Again, in the same website, they have also given some checklist. Okay, and that checklist can help us in preparation for the practical classes better than what we are doing at present. Okay. So whatever I'm going to share in this 4 or 5 next slides is the extract of this particular website, where I feel that the learning objectives have been very nicely given, and that can help us all as a very nice tool for pedagogy. Okay. I'm taking you again to my presentation. So friends, this is the background. And by now, I think you people know what is the definition of learning objectives. Okay. Again, may I request someone to tell what is meant by learning objectives, so that there will be some, I can understand how much you have gone through this particular definition. Any one of you, please. Teachers, what is meant by learning objective is my question. Yes, anyone can unmute and tell, please.
I think Dr. Suhas wants to answer.
Yeah,
He has
please,
unmuted
please.
himself.
Please, Dr. Suhas. Yeah, please participate in this discussion so that we can go ahead rapidly.
Dr. Suhas, you want to tell the answer? Or maybe Mr. Vivek?
Yeah, Mr. Vivek.
Yes, ma'am.
Yes,
Yeah.
sir.
Yes.
Please.
Hello.
Yeah, what is
My
meant by learning objectives?
use is nothing but after completion of that particular course or practical, they should understand few things, what is important in that perspective.
Right, okay. So this is a rough definition of learning objectives, but if you go to any pedagogy related book or research article, Dr. Kalpesh Gaur, do you want to add something?
Sir, I want
Yes,
to
sir.
Yeah, Dr. Kalpesh.
Learning
Please.
objectives should be if clear-cut statement of what will be able to do by the
Right.
end of the lesson.
Exactly. So we are coming to the very nice definition of these learning objectives. Learning objectives are statements which define about the measurable outcomes of a teaching plan. Okay, this is something which happens while drafting the learning objectives. Means when a teacher is planning for his class, he is first writing down the learning objectives for his students. And if you see the statements, even this is there in the PCI syllabus, that this learning objectives start with something like this: after listening to this lecture or after performing this practical, students will be able to. Okay, and then there is a list of different factors which we are expecting from the students. That is what is learning objectives, and this have been given for theory classes. That's what I'm repeating again. But for practical classes, there is nothing like learning objectives which have been mentioned. So in this particular discussion, we're going to plan out what should be learning objectives for the practical classes, and then we'll be going for online or blended or hybrid kind of practical classes in present scenario. Okay, so friends, I had taken you to Leicester University website. I told you that from there I've copied down some objectives which I feel are really ideal. Okay, so in the Leicester University website, for practical classes particularly, it has been said that after attending the practical classes, the learner will be able to consolidate subject knowledge. Learner will be able to introduce disciplinary methods and procedures. The learner will be able to develop technical skills. He will be able to develop cognitive skills. He will be able to acquire teamwork skills. He will be having increased motivation and interest in the subject. So whenever we are planning for a practical class, this is what we are actually doing. Okay. Now, I am sure that out of these 100 teachers, all of us are unconsciously doing all these things. Okay. but we have never written down all these points for our own references. So that's what we can do through this particular discussion, that whenever we are planning a practical class, we want our students to do all these things. Again, question comes, is it really not possible to do all these things in the theory classes? Why we need practical classes then? Friends, if you look at these particular objectives properly, then you will be easily understanding that we cannot attain all these learning objectives in the theory classes. Okay, objectives for theory classes will be a different set of objectives. For practical classes, it is very important that we have a clear-cut idea of why we are conducting that practical. Okay, so consolidating the subject knowledge is the first thing that we do in practicals, because see, students have already attended the theory class, they have learned something in the theory classes. Now we are bringing them to wet laboratories, and there we are helping them to consolidate what they have learned, to confirm what they have learned, okay? To assimilate what they have learned. For that particular reason, we call the students to practical classes. Why I am going up to these micro things? It is because I want to let you know that it is not really possible to conduct online practical classes. And then I'm going to tell you something about the ways in which we can enrich our practical classes by using certain resources which are available with us. Okay, so just bear with this explanation that I'm dealing with in the beginning. In practical classes, we are enabling students to consolidate the subject knowledge, because there we are interacting with the students for 4 hours together. Okay, in our lectures we do it for 45 to 50 minutes, but in practical, students are there with us for 4 long hours, and that is how we are trying to consolidate what the students have learned in the theory classes. Okay, now let me share with you that due to corona now, what people are doing is they have reduced the duration of practical classes to just 2 hours. Okay, why? Because teachers complain that we cannot use Zoom, or we cannot keep on talking with the students for 4 long hours. That is the reason why we should reduce the duration of practical classes. Is there someone in whose institute this particular strategy has been accepted? They just reduce the duration of practicals. You can just mention in chat box if something like this has happened. In many organizations now, people have just reduced the duration of practical classes because they have to conduct practicals online. Okay.
People are responding, sir.
Yeah, people are responding. I'm thankful to everyone who is responding, okay? Because what happens is, if it is a one-way kind of interaction, then it won't be that interesting. Okay, so
Yes.
2-3 of them have
Said yes.
said that yes, this is the strategy which people are using for online practicals. Don't you feel this is really unfortunate? See, like teachers, we should first think about the learning objectives that we are not doing. Then, instead of enabling the students to go ahead with the advantages of practical class, we are reducing the duration of practical classes.
Absolutely.
Yeah, and we are saying that this is what is necessary in the era of COVID. Okay. Question is, within 30 to 45 minutes, is it really possible to meet learning objectives of practicals? It is highly impossible. See, whoever is accepting this kind of decrease in the duration of practicals, they are not looking at these learning objectives properly. Okay, so if suppose you want to conduct a practical online, you should plan it for all 4 hours. Now, I'm sure people will laugh at me because staying online for 4 hours, interacting with the students for 4 hours is something ridiculous, right? I'm not telling something like that to you people. I'm telling that you can plan this practical related interactions in such a way that all 4 hours students will be engaged in learning, and they will be able to achieve the learning objectives related to the practical classes. I think now I have highlighted what is the serious issue related to our understanding about practicals. Okay. We easily shifted towards reduced duration of practicals because we did not know why we are conducting practicals, or we were not told systematically what are learning objectives of the practical classes. Okay, so friends, this is an effort to sensitize all of us, including myself, okay, because these things are happening in my organization also today. Today we are conducting practicals just for 2 hours. We keep Zoom session of 2 hours for the practical, and we try to deal with the practicals in whatever possible way we can do it, by using YouTube videos or some showing something to the students in live mode or something like that. Okay, but is it really doing sufficient justice to the learning objectives? That is the big question which all of us as teachers should answer. Okay, now the first objective is we are trying to consolidate the knowledge that students have learned earlier in the theory classes. If we keep this one objective in our mind, we can plan our this whole practical, 4 hours practical, considering what we want the students to revise. That should be the first part of our practical class. Okay, no doubt you people may say that this is too much ideal, who will be there spending 4 hours in this corona day and all. But friends, if we consider these things sincerely, we can plan a 4 hours practical, engaging students into real learning experiences and enabling them to meet all the learning objectives. Okay, so as per the Leicester University guidelines, the first objective, that is to consolidate the students' knowledge. So if you plan for this, you can enrich your practical classes, and you can prolong those practical classes, or you can give sufficient time for the practical classes. Okay, let me take you to the second objective of practical class now. That is to introduce the students to disciplinary methods and procedures. Okay, friends, I am a pharmacology teacher, and if I see the syllabus of PCI for pharmacology, there are certain experiments like toxicity testing as per OECD guidelines. Okay, in I think pharmacology 2 or 3, this practical is there. There is a practical for skin irritancy testing. Okay, let me ask the teachers, pharmacology teachers here, have you conducted these practicals? Is there someone who has conducted toxicity testing as per OECD guideline? Please, teachers, respond to my question. You can just post it in chat. 1 or 2 of you, or if suppose you are just skipped that practical, say that you skipped the practical. I want to know about this. Then only I will be able to tell you how to conduct that particular practical properly. Out of 4 or 5 pharmacology teachers, is there someone who has in last 6 or 8 months conducted the practical of toxicity testing or the practical of skin irritancy testing? These practicals are there in PCI syllabus of pharmacology 2 and 3.
Can anyone please respond to this?
I'm asking about specific 2 practicals, toxicity testing as per OECD guideline 425 and skin irritancy testing as per OECD guideline. Is there someone who can add to this, please?
I guess, sir, the answer should be no, because nobody is responding.
Right, nobody is answering.
Yeah.
All right, so let's consider that no one of us has taught those practicals online. Okay.
Yes.
So I'm happy that I'll be able to share with them how to conduct that particular practical today, right? By using certain online free resources. Okay. Now, friends, let me take you again back to this particular second objective. See, all of us are teachers, and I repeatedly request your attention to what I'm talking about, because lack of learning objectives and not even a single teacher trying to define the learning objectives is a serious problem, I will say, at the national scale. Okay, all pharmacy teachers, someone must have come up with a suggestion to PCI that no, we should include proper learning objective for practical classes also, which has not happened. Okay, in last 3, 4, 5 years, no one really thought that this should be done. Okay, but now If it is not there in the PCI syllabus, at least at personal level, we should look at these things seriously. Then only we will be able to give justice to our students regarding practical classes. Okay, and our responsibility has increased because of Corona. In the time of Corona, our students are very badly deprived from the real subject matter. That is where, as teachers, we are supposed to contribute to their future. Okay, that's what I'm trying to highlight here again. So through practical classes, first objective I told, it is to consolidate their knowledge. Second objective, learning objective for the practical class is to introduce them to the disciplinary methods and procedures. That is the reason why I mentioned about these two experiments particularly. Toxicity testing, pharmacologists have to do. They must be aware about the whole procedure. They must be able to perform that practical and tell whether the compound is toxic or not. For that, as teachers, we need some resources. Okay, and we must be seriously looking at this particular practical now. Let's look at the third objective. Okay, again, these are objectives of practical classes particularly. We want our students to develop technical skills through practical classes. So whenever the next online practical you will be conducting, you just ask yourself as a teacher, am I teaching my students about the technical skills? Is my student developing technical skills by listening to me? At least cognizance related, cognitive skills which are there, is my student achieving them? This is a serious question which we teachers should ask ourselves. The purpose of putting practicals in the syllabus is to provide technical skills to the students. And online learning is never going to help for this. Okay. Now we must highlight this thing and then try to prepare the study material in such a way that at least the students will be having some feeling of technical skill that is necessary to perform that particular experiment. Okay, so you can imagine the tremendous responsibility that is there on the teacher's shoulders today to enable students to develop technical skills by looking at or interacting with some online material. To that extent, the problem is serious. Okay. Let us look at the next learning objective, that is to develop cognitive skills. Okay. In practicals particularly, we are providing students with situations where their cognitive skills will be developed, where their creativity will be developed, where their analytical skills will be developed, where they will be able to interpret the data that is recorded from an experiment. So friends, within 30 to 45 minutes, do you feel that you can enable a student to analyze the data? My question is, don't you have a data set which you can present to the students and ask them to make some inferences out of it? Can't we bring such innovative techniques to engage our students and attract their attention to what actual cognitive skills they have to develop through the practical class? As we are not having this set of objectives, we are not looking at these things. But friends, I'm sure that if suppose we teachers look at learning objectives of the practical classes properly, I'm sure that we will be planning our practical classes, online practical classes very, very nicely. And I'm sure that all of us have that capacity. All of us can design the practical classes in the best possible manner, even by using online methods. Even through Zoom, you can conduct a very nice practical class where the students will get complete feel of that experiment and all learning objectives will be attained by the students. Okay. Friends, apart from all this cognition, technical skill, consolidation of knowledge, there is something very important that happens in the practical classes, and that is teamwork. Okay. Now, I'm sure the listeners will be slowly smiling at me because I'm talking about teamwork while a teacher is discussing something on Zoom. Okay. You can ask me whether it is really possible to carry out teamwork in practical classes. I mean, can we make our students to perform a teamwork? But friends, there are methods to enable our students to perform even teamworks during the practical hours. Okay, So we have to look at the possible resources in this particular manner. Okay. So this is the very important aspect that we pharmacy teachers have thoroughly neglected. We are living with a syllabus that has no learning objectives for practical classes. That's fine. We are moving to online mode. There we are totally neglecting again whatever was at least we were doing in the practical classes. Okay, so all these things are quite serious from the side of on the side of teachers, and they're doing serious harm to the students also, because students will just look at the list of practicals. They'll try to write something in their journals, they'll submit the journal, and they will pass out. But that is a serious harm that we are doing to their technical abilities. Okay, so friends, the last objective of practical classes is to inculcate teamwork amongst the students, create the values of ethics, enable the students to collaborate with each other, teach them for team building, motivate them, ask them to focus on the study material, and so on. Okay, and then students must provide constructive support to each other through the practical classes. So these very broad, very ideal objectives are there behind each practical. Okay. So when we are looking at practicals henceforth, I request all the teachers to just keep this list of learning objectives in front of them and then design the practical. Let it be online or offline, but at least we should know that these are the learning objectives which we are supposed to look at when we're planning our practicals. Friends, thanks a lot for looking at these objectives very patiently. I hope all of you at least felt that somewhere these things must have come across, we must have come across these things earlier, so that we could have looked at our practical classes more efficiently. Okay. A small question, a reply I will expect from at least a couple of people from audience now. Do you feel, friends, that these learning objectives are important? Yes, please share with me. You can unmute and share, or you can post in chat box. I want your responses, please.
Sir, hello.
Yeah, please. Yeah, Dr.
Sir, actually, I have a question.
Yeah, please go ahead.
Sir, my question is that for the labs, actually, in our labs, what do we do during this lockdown? What we have done is that we conduct practice, done the practicals by ourselves, recorded the videos, and show them to the students, or use streaming. But the question is, until and unless actually they are performing it by hand, so some gap should be there.
Right. We are accepting
and
that
also,
gap
sir,
is
in
going
some
to be there.
cases, like,
yeah, please.
And also for certain subjects like pharmaceutical biotechnology, previously there was a practical.
Right.
But now in the new syllabus, with the practical is not there. So without going through all these practicals in biotechnology, so this subject will be difficult to understand.
Exactly. So thanks, Professor Ghosh, for sharing that these problems are there. My question, I'm going to talk about these also, the aspects which you have talked. But Professor Ghosh, please unmute yourself and tell whether these learning objectives can enrich our way of looking at the syllabus or not. Professor Ghosh.
Hello.
Yeah. My question is.
Yes, sir.
My question
yes,
is.
Sir.
Had you thought about these learning objectives?
Objective will always help.
Right. Thank you. But you had not looked at these learning objectives.
You have to fulfill, right?
Right. No, but while looking at, while preparing those videos, had you thought of these learning objectives?
Sir, I would like to add.
Yeah,
actually, it's like you mentioned teamwork also. This never came in our mind that with our Zoom sessions, we can ever think of inculcating teamwork in students. So we are really eager to listen. How can we actually inculcate this quality?
Yeah. So friends, this is what I wanted to tell you, that there are methods. See, I'm not saying that through Zoom only you will be able to develop teamwork. You can develop teamwork by using some another platform where you will be giving a common problem to the students to solve. There are few examples I'm going to share with you. That was the background on which I wanted to start the demonstrations. Couple of demonstrations, 3-4 demonstrations are there. But the whole purpose of telling these learning objectives is to let you know that these many things are expected from us as teachers. Okay. And when I am a pharmacology teacher, I have looked at pharmacology syllabus from my angles. No doubt others also can look at their own subject domains for such kind of resources. Okay. And the question that people will ask is, okay, pharmacology आप लोगों को तो online पढ़ाना है because it is a software based subject. Pharmaceutics में क्या करेंगे? Biopharmaceutics में क्या करेंगे? All these questions are relevant. Okay. But friends, if we start looking at resources, it is sure going to help us. That is what is my stand. And that's what I am going to share with you in today's demonstrations also. Okay. So all teachers, we sure accept that all the practicals we cannot demonstrate, all the practicals we cannot conduct in online mode efficiently. This is underlined. Okay. This is the bottom line. All practicals we cannot teach by using online methods, but whatever we are doing in online mode, that we can enrich if we keep this list of learning objectives in front of us, and if we look at the resources properly. Okay. The problem with us is we are not looking at the resources on internet is full of hundreds and thousands of such resources which can be implemented for practical classes. Okay. I'll be showing few examples related to pharmacology. Okay. So first thing is, when we are saying online learning, it is not going to fulfill all the objectives, but we can enrich the way in which we are conducting practicals online. Okay. At least there are few experiments in our syllabus which are demonstration related experiments, okay, or which we carry out as demonstrations. If suppose there is HPLC, then at degree level we are not providing each student with HPLC facility. We are not giving access to each student. But there are possibilities that you will be having such a nice interactive software which will enable the student to actually solve the problems related to HPLC or HPLC, say peak isolations or peak separations and all. That kind of softwares we have to look at. So it is important that we should start using those online resources efficiently to make even our conventional teaching learning process very effective. So let me take a couple more minutes on this particular slide, because here I am not concerned only about COVID situation. This COVID will go up to a couple of months or few months. Once the vaccine comes, I am sure that we are going to forget about Zoom. Okay. And we are going to continue the same routine which we were using before a year. Students will be coming to laboratory, they will be performing practicals, they will be writing the results, journal submissions, so on. All those things will be there. Again, the question is, was that method effective? Can we say that we are really planning out practical classes to meet all these learning objectives, or had we thought about learning objectives at all while designing our practicals? Okay. Friends, that is where pedagogy comes in picture. That is where blended learning comes in picture. Okay. For practicals also, we must utilize some online resources efficiently, and we must have our own design of practicals considering these learning objectives. Okay. So this is where blended learning is coming in picture. This is where hybrid learning is coming in picture. Okay. So let's have a look at few resources which are there for pharmacology subject, at least pharmacology and allied subjects. Okay, last time also I had asked this particular question: how many of you got access to, or how many of you have used this very nice resource for conducting practicals? In today's session again, please post in the chat box whether you have used this particular resource. Any of you? Yeah, yes, I got 1 and 2. No one is responding yet. How many of you use this particular resource? I want to know.
Sir, I have used it.
Yeah, first, thank you for human anatomy and physiology practical. Thanks a lot, ma'am. Is there anyone who has used this for any other practical?
Sir, I have used it for biochemistry.
Biochemistry,
And for
exactly.
microbiology also I have used it.
Yeah, very nice, madam. Thanks for sharing this. So, audience, you please note that our colleagues are using this very nice resource for conducting anatomy, physiology, biochemistry, and microbiology practicals. This is really a great resource. I hope each one of you should use this. Can we use it for HAP? That's a great question, Mr. Professor Vivek. Yes, we can use it for HAP. Just last week, I used a part of this particular software for conducting my class. Okay, let me take you to the this particular website now. Hope you can see the screen.
Yes, we can, sir.
Yeah. So this is the website which is having, I think, almost 100 experiments in it, in which there are anatomy, physiology, molecular biology, biochemistry related experiments. Let me show you a sample of this. There is an experiment on hemocytometer. Okay, for first year HAP, we're having this practical. Okay, and in last week itself, I used this particular platform to tell students about how to use microscope, how to look at the cells, how to count the cells. Okay, so all this thing the students can do on their mobile screens using this particular and very informative, very nice resource. Okay, see what you are looking at on this on your screen now is a microscope under which this Newbus chamber has been highlighted. Okay, now the beauty of this particular resource is you can move the slide by using this particular controls which are there. Okay, then you can focus it by using high power and low power. Can you see that? So students will be having exact feel that they are looking at a slide Newbus chamber filled with suspension of WBCs. Okay, and then you can ask them to count the cells which are present in each of the squares, enter those values here, and they will be getting a total. Okay, now friends, my simple question is, if suppose you use this particular resource, don't you feel that your students will be at least understanding what is really being done in the practical of WBC counts? Okay, then you can tell them about the calculations which are involved in this. Then you tell them why WBC count is important. Then you ask them whether there are any diseases related to WBC count. Okay, so this is how, see, I was using my 2 hours time to explain about this simple practical. And then a part of it I had to leave aside because the practical duration which was given of 2 hours, it was over. The students had to shift to another lecture. Okay, so this is what I want to tell you. If you look at the resources like this and then plan your practical in such a way that your students will be having the real feel of looking through a microscope, adjusting the details, moving fine and coarse adjustments, and then count the cells, write them, and take the total count. Okay, then on screen you can show them how the Newbus chamber appears. Then you can actually hold the Newbus chamber in front of your mobile or this laptop camera, show them how it is etched with different markings. Then on screen, by using this Zoom related application, you can show them whiteboard particularly. You can show them how that calculation has been done. You can ask them to solve particular thing related to uh, determination of volume and all. Okay, so this is what I did just last week. I started with just showing them a Newbus chamber. I held it like this in front of the camera. Okay, and show them that this is something which is called as Newbus chamber. Then I started with the types of WBCs, why WBC count is important. I told them about the dilution fluid, constituents of dilution fluid, calculations related to dilutions, and so on. Okay. So if we really use these resources, and mind well, this particular resource is having chemistry, biotechnology, molecular biology, many type of experiments are there, and they are freely available. That is the beauty of this particular resource. Okay. Our government has funded for free distribution of this software. Just you have to register to that, get access to it, and start working on that. Okay. There are many aspects, even in theory classes, which you can explain by using this very nice resource, which is open educational resource. Okay. And this open educational resource is something that is a revolution in the field of teaching learning process. Okay. You will wonder that even UNICEF, the international organization UNICEF, has got funding for development of these OERs, or it is funding these OERs. Okay, someone has asked whether it is really free. Yes, friends, it is really free of cost, totally free. Okay, you can use it for your routine classes without asking anyone. Okay, that is the beauty of this resource. That is the beauty of OERs. Okay, and then if you use the videos of this particular resource, you need not to take any permission from anyone. For your classes, you can very happily use all these resources. Plan your practical using these things. That is the very important aspect of designing our practicals. Okay. Then I am shifting rapidly to the next resource. Okay. You people can get access to this and go through it. Okay, it is free of cost. You can use it for n number of hours. Another thing is there, again in pharmacology practical. There is one practical, I think in pharmacology 3, where PK/PD parameter calculations or pharmacokinetic parameter calculations have been mentioned. Okay, we can understand that using actual animals or even human related data, we cannot explain all these things to our students. But if you are using another free resource, mind well, this is also a free resource, interactive clinical pharmacology. If you search for this in Google, you can get access to this. Here you even do not need to register. It will directly start. Okay. Let me take you to this particular resource. This is for practical of pharmacology, where pharmacokinetic parameters are to be explained to the students. Okay. All those parameters have been very nicely presented in interactive manner in this interactive clinical pharmacology. You have to just go to icp.org.nz, okay? And there you will be getting all these details. Just let me take you to one of these interactive demonstrations. See here, if you want to explain to your students the concept of volume of distribution, you can ask your student to just open this particular software, ask them to inject the drug here. If the drug is distributed like this, volume of distribution will be 1 liter, like this 10 liter, like this 100 liters. Yeah, this is simplest thing, but all the concepts of drug distribution along with their calculations have been shown here. Okay, so if you want to tell your students how actually hepatic elimination, renal eliminations are related to each other, you can use these particular sliders and tell your students how it is happening, or how they are correlated. Okay, if you want to find out bioavailability, liver function, if suppose liver function goes down, what happens to bioavailability? If suppose renal function goes down, what happens to clearance? All these things you can very nicely represent to your students, and they will be getting the real idea, or at least instead of talking in front of students using some stable images, such kind of interactions can enable your students to understand the details properly. Dependent upon this, now you can plan for your interactive sessions during practical or after practical. Okay, so see, in this very nice resource, many things have been explained, many things are easily demonstrable. The beauty is it is interactive. Okay, so if suppose you are putting some drug, if suppose there is first pass metabolism, how the things are going to change, all these aspects you can demonstrate by using this particular thing, and then you can share some data sets with your students during the practical hours itself. I'm sure if you're using such kind of resources, 4 hours time will be insufficient to explain the details to students. Okay, and you will be feeling the satisfaction at the end of your practical that yes, you have contributed something to the students' understanding. Okay. So this is another resource which you can use to teach a practical of pharmacokinetic parameters, which is there in Pharmacology 3 syllabus of B Pharmacy. Okay. I'm tempted to ask the participants again, how many of you have come across this resource at least before this? Is there someone, even pharmaceutics people can use this particular resource? So I want answer from pharmaceutics people also now. Have you ever used this interactive clinical pharmacology for theory or practical classes? Have you shared this information with your students?
Faculty members, please do answer this.
No.
If we have any
Yes.
pharmaceutics, no.
Yeah.
Yeah, from Dr.
Okay,
Sumas.
let's take it as a representative example that these things have not been used.
Yeah.
Thank you, Professor Sakarkar, for interacting. Okay. Professor Ragib is there.
Yeah.
Sakarkar, sir.
So all are saying no.
Yeah, right. So friends, even though this is a simple free resource, I think students will love to interact with this particular resource. And yeah, someone has said it. Someone has said we were not knowing. Okay, I thought it was not working. No problem. Okay, friends. So this very nice resource is there. See, this is something which I found out while surfing through the net. Okay, I am sure there are hundreds of such resources available. Okay, so in one of the earlier conferences, I conducted similar lecture, and then a pharmaceutics professor asked me, सर हमारे यहाँ पे ये नहीं होता है, लेकिन हमें कैलकुलेशन समझाना है स्टूडेंट्स को, उसके लिए कुछ है क्या? At that particular moment, I had not seen on Google whether there is something for this. But friends, multiple interactive calculation softwares are openly available, freely available to us, using which we can make our practicals interesting. We can make our teaching interesting. Okay? आप लोग सिर्फ स्टूडेंट्स को इसके वैल्यूज दे सकते हैं, सैम्पल वैल्यूज, वो साइट पे डाल के उसमें एंटर करके वैल्यूज स्टूडेंट्स समझ सकते हैं कि हो क्या रहा है उसमें. Like this, multiple resources are available, free of cost resources are available. Okay? And this kind of OERs which are there, they are mind-blowing really. If you start using it, you will get addicted to them really. Okay? To that extent, they are nice. And students will also feel something new, that they are having some interactions and they are trying to understand the things. Okay. Let me rapidly take you to the next resource, which is again from pharmacology. Friends, this is the question which I had asked. And at least out of this sample of 100 or 100 teachers, I'm sure no one has used this particular software for the practicals. If there is someone, please raise hand or post in chat box. Even this software is freely available on internet. It is available on OECD website, using which you can demonstrate the practical of toxicity testing as per OECD guideline. Okay, and your students can have the feel of using exactly standard software which is used at international level, which is used even in industries. Okay. Using this, your students can calculate the values for toxicity testing or LD50. They can calculate by using acute oral toxicity 455 guideline. Just look at this. See, the student has to enter this name of the animal, whether the animal died or the animal survived. Okay. The software will suggest that the next dose should be 55. So next animal you have to dose with 55. See whether the animal dies or not. I am saying the animal did not die. Here also the animal did not die. Again, the software is suggesting go for 175. I am entering 175 dose. I'll say that the animal died at this dose. Animal died at this dose, even as a long term. Then again, the next dose is suggested. Just yep. Yeah, is there any problem? Someone has, I think it was by mistake. Okay.
Yes, sir.
Yeah. So, like this, see, your students will be interacting with this software. They will be putting these doses. See, now the software has said that you have to continue dosing here. The animal is living alive. Animal is living alive. And then, okay. Now, the software is telling us that the test is complete and estimated LD50 is 98.11. Friends, you can ask your students to download this particular software on their mobiles and ask them to go through the entire process. Then you give them some challenges. Why this particular value is not coming, what is the rule, stopping rule for this particular criteria, and so on. Okay. Even this is freely available, free of cost totally. And it is present in OECD guideline 425 on OECD website. As the practical is there, it is our duty to try to explain it the best possible manner to our students. Okay. In this, then again, you can see literally if you want to teach this practical, you will need more than 4 hours for B.Pharm students. Unfortunately, we are reducing the time of practicals. Okay, but if you are having these resources, if you are looking for these resources, 4 hours are also insufficient. Okay. So that is the beauty of this open online resources that we are having. Okay. So this was about the third or fourth practical that I am telling you. Okay. Madam, can I have 10 more minutes?
Please, most welcome, sir.
Yeah, thank you. So friends, we are going to now the next resource. This is for theory class. I am not going into details of this, but for anatomy physiology, you can use this particular website to enrich your students' learning experience. This is from Carnegie Mellon University, a total open source course for anatomy physiology and many other subjects. This is freely available. Even during your practical classes, you can ask your students to spend 1 hour on this particular content detail, ask them to go through a quiz which is provided there, help them to solve the quiz properly. That will really help the students to understand what they are learning in the classes. Okay. Here you just have to register for this website, get access to the very beautiful contents developed by experts, and this is a global standards, we can say. Okay. You can visit this course. There are very nice presentations, questions, everything is there. Okay. Explanations are there. So your students will be, I'm sure your students will love to see all these things. Okay. Even this is free of cost. So in practical classes, see, there are questions. Then there are resource materials available for this. Much interactive content has been given in this particular website. Okay. So you can use these things for enriching your practical classes. Let me go to the next part of it. This is where at institutional level we can request our managements to subscribe for this Bodhi tree. It has been developed by IIT Mumbai. Okay. And in many conferences where I have been a resource person, I have been telling people to just contact these people of Bodhi tree, because they are charging 5 rupees per semester per student. Okay. You can see the cost. And then you can ask why 5 rupees? Because they want to pay for the server, that's all. So this is totally an academic project which has been successfully completed by IIT Mumbai. They are providing this LMS, Learning Management System, free of cost, in which we can develop our resources and ask the students to take benefit of this. Okay, I'm taking you to a small practical. It is not actually a practical, but a small content that I have developed by using this platform, where I took a training of this Bodhi tree for a week. Okay. I'm going to search for pharmacology. In pharmacology, just now I mentioned that there is a practical named skin irritancy testing. Okay. Now the beauty of this particular platform is you can create your multimedia book. Okay. And this multimedia book students can access through their username and password on using their mobile phones. This Bodhi tree comes as an app. Students will be having their own username and password. You can enroll the students in this particular course or the material that you are developing and enable them to understand the practicals or theoretical aspects, or even you can ask them to appear for the test and all those things you can do in a very, very simplest manner. You will not need any software related knowledge to do that. Once if you see how to develop, it is quite easy. Even videos you can use in the best possible manner. Okay. So the skin irritancy testing is a challenging practical for degree classes, where if suppose the teacher is unaware about this resource, he will not be able to demonstrate that practical. Okay. Actually, for this skin irritancy testing, there is a free resource available on internet. There is a YouTube video. Now you can ask me what I can do using that YouTube video. Friends, there are free of cost resources using which you can create your own learning videos without spending even a single rupee. Okay. Just have a look at this particular video, which I have created to tell about this skin irritancy practical. Okay. I'm sharing this sound now. I hope you can hear this video now.
Yes, sir.
Okay. This is for just demonstration purpose. I have to prepared how to demonstrate to our students a very advanced kind of pharmacological experiment, which is there in their syllabus now. This also is there in PCI syllabus of pharmacology. Okay.
Hello everyone. This is one resource which you can get free of cost on the internet, which tells about episkin, human epidermis grow in a petri dish. Okay, you can use this human epidermis, which has many characteristics similar to human skin, and you can test your compounds directly on this. Okay, now while doing this practical, or while explaining this practical, you can ask the students to watch this video. In this video, you can embed the questions. Okay, like this, like the one question that I have embedded here.
See, now the beauty of this particular resource is student will not be able to see the further contents of this video unless and until he answers this particular thing. Okay, and such questions you can embed in the videos. See, now as a student I am answering this question, I will submit this, and if I am correct, the next video or video will be playing after that. If
And
I am not
students
correct,
will not be able to see the next part of this video unless and until they see this particular question and they solve this question. Like this, we can create our own videos using open source material on the internet, put them in your learning management system, which is again very cheaply available, and you can demonstrate actual experiment to the students as if they are performing it in, or they are seeing the demonstration in wet lab. Okay. So all these strengths are there of this particular learning management system. Without spending even a single penny, you can create such videos.
okay. So friends, I think you saw the beauty of this kind of learning management systems, the practicals which are there, which cannot be as such demonstrated, but for them some resources are available. You can create such nice videos for your own practical classes. You can embed the questions, you can make your students to see the entire video. Unless and until they see that video, they will not be able to answer the question. And unless and until they answer the question, they will not be able to see the next part of that video. Even such kind of interactions have been created. Means teachers have been we can say they have been empowered to create such resources which are compulsorily interactive. Okay, so your students cannot skip what you want to tell them. Similarly, there is another resource called as SAFE, which is for conducting examinations. This resource is also available with Bodhi tree, where you can carry out examinations in a remote mode, which enables the students to give attendance, you can observe the students in between, and you can say you can more confidently conduct the online examinations. So these two resources have been created by IIT Mumbai. And if you just look at the email ID which is there on this website, Bodhi, you can communicate with them. They are very helpful people. Actually, this is a pedagogy related research project. Okay, they already developed it for teachers, considering their needs, teachers' needs, and then this can be purchased for a institute just at 4,000, 5,000 rupees. That's all. Or even an individual teacher can subscribe for this just by paying 500 rupees. That's all. Okay, so this is how you can make your teaching interactive. And in this Bodhi tree like aspect, or if suppose you're using a web, what we say WordPress kind of website, which is easy to develop, you can create groups among the students who will be learning the things together. Or you can create some challenges for the students where their creativity will be coming into picture, and then they will be submitting their particular answers to that question. This is how you can use all these resources. See, this is what I have explored on my own. But if suppose 100 teachers start exploring, you can imagine how beautifully we can teach the practical step. And if 100 teachers come together and they develop such a nice resource, I don't feel we will be having anything shorter than any advanced country in the world. Okay, but this is what we have to look at. For this, we need a proper understanding of learning objectives. Then, friends, these are the resources which I had created through my rigorous hard work of 7 to 8 years. Very beautiful presentations I have created. Then there are interactions students can look into, or students can interact with this. Using these resources, students can even take their exams. You can conduct systematic examinations of students by using these simulations. So regarding this, I'm not going to talk too much. I hope majority of you have seen this particular thing. If you're not seen, I can share this presentation with the organizer so that you can get it from the organizer. Just click on these icons. You'll be getting the YouTube presentations or YouTube videos for these demonstrations. Okay. So this is what I had brought for you today. I'm thankful that the session was sufficiently interactive. And thanks to everyone who has responded to my questions. Okay, it made me continue with some more enthusiasm. Last time it was means people were not at all answering anything. So
Last
compared to
time
that,
we had separate set of people. This time we have
right.
some new ones.
Yeah, I'm happy that they responded. Thank you. Thanks a lot for patience.
Sir, thank you so much, sir. It was really a very informative session. In fact, I should say in this pandemic, we all are facing the issue of how to conduct practicals.
Right,
so you have made it so simple and easy.
Yeah,
and I would request if anybody wants to ask sir something before
please.
sir leaves. Anybody who wants to ask can unmute also and ask. Or you can put it in the chat box, whatever way you're comfortable. One Dr. Ragib is asking if you can share all the links which you had just.
That's what I'm going to share this presentation with the teachers.
Okay, Chitra ma'am wants to know, can you apprise us of any resources for biopharm and pharmaceutical practicals?
Yeah, that's what, madam. We will have to start looking at Google. That's all. We'll get it. See, for pharmacology, we've got such nice resources. For other subjects, also we'll get. I'm sure.
Right. But the best part is, sir, most of the links which you showed were all free. So
yeah,
we
all
can.
Of them are free. Yeah.
Right. So thank you so much, sir. Anybody else who wants to ask? Pratiksha Mishra, she has raised her hand.
Yeah, please.
So I guess she wants to ask something. Miss Pratiksha? I think it was by mistake.
Okay.
Yes. Okay. Sir, Dr. Pradeep is asking, where can we purchase these softwares? But you just said they are free.
See, last software which was there of pharmacology.
Yes.
Simulation, that is a paid software.
Okay.
Otherwise, all other are free.
Okay. Right. So I hope Pradeep sir, you must have got your answer. So sir, you can just share, if you can share us the links later, we can share it with the.
Yeah. I'll be sharing this presentation with you, PPT with
Thank you, ma'am. So thank you so much.
Goel sir has raised his hand again.
Dr. Pradeep Goel, would you like to ask something?
Again, Anup Kumar is asking, is this software free version?
Yeah.
Okay, see there, don't mix these two things. The last slide, which I did not explain too much, was my own software that is available in market as paid software.
Okay.
Earlier, all resources which I have shown you are free of cost.
Okay, so I hope it is clear to everybody. Only the last one was a paid software which has been created by sir. Others are free of cost and they are available in the internet.
Yeah, and I'm sharing this presentation with you all,
yes,
so
sir.
that you can just have those links directly.
Right. So you can share it with Chitra ma'am so that we
I'm
can
sharing.
extract it from
I'm
there.
sharing.
Yeah. Thank you so much, sir. And I'll just share my screen.
Yeah, please.
Thank you so much, sir. And please accept this e-certificate
Yeah,
from our side. Thank you
Thanks
very,
a lot.
very much.
Thank
And
you.
hoping to see you again in our future FDPs as well.
Okay. If people are liking this, then I would
Sir,
like
we
to
are liking it
participate
very
more.
much. In fact, you can see the number also. It is more than 100.
Yeah. Thank you.
Yeah.
Thank you.
Thank you, sir.
Sir, I would
Yeah.
like to add, it's a
Yeah,
personal thanks on my behalf that on my
Okay.
request that you made it for today's session in spite of your busy schedule.
Yeah, thanks
Thank
a
you
lot.
so much, sir.
Yeah, thank you.
Thank you very much.
Thank you, sir.
Yeah.
Thank you.
Okay.